Welcome, everyone. Thank you for joining us for today's webinar. My name is George Carrick. I'm the Director of Marketing at Sinclair Research, and I'll be your moderator today. Sinclair Research has emerged as a mid-sized non-clinical CRO offering a full range of in vivo non-GLP and GLP research services in rodent and non-rodent animal models, including IND-enabling toxicology and safety pharmacology studies to help progress your compound from late-stage discovery to phase 1 clinical trials. You can learn more about Sinclair's IND-enabling research services at sinclairresearch.com. XenoTech is a specialty global CRO specializing in ADME, DMPK, and DDI non-clinical testing of potential drug candidates. As a premier provider of in vitro drug metabolism and drug-to-drug interaction studies to pharmaceutical companies worldwide, XenoTech offers sponsors unparalleled experience in evaluating drug candidates as substrates, inhibitors, and inducers of drug metabolizing enzymes and drug transporters. You can learn more about XenoTech's preclinical in vitro drug development services at XenoTech.com. Today we're presenting Selecting the Right Species for My Small Molecule Program, the Role of In Vitro Assays, presented by Scott Boley, Senior Vice President of Research at Sinclair Research, and Andrew Taylor, Manager of Technical Support Services at XenoTech. Today's presentation will dive into the scientific rationale driving species selection for your non-clinical program, with an emphasis on small molecule programs and how in vitro species comparison studies can help determine the right species for your in vivo program. Now let's meet our presenters. Dr. Scott Boley has advised sponsors on their non-clinical needs for the last 15 years and managed toxicology programs for 19 years. He has extensive expertise in drug development, as well as the regulatory expectations for a variety of test article types and indications. In his role as Senior Vice President of Research, Scott oversees the research, report services, and send groups at Sinclair Research that support each sponsor's non-clinical safety programs. Scott is a diplomate of the American Board of Toxicology and holds a Bachelor's of Science in Biochemistry and a Ph.D. in Biochemistry and Environmental Toxicology from Michigan State University. Dr. Andrew G. Taylor received his Ph.D. from University of California, San Diego. He joined XenoTech as a research scientist in 2017, serving as a study director in non-clinical drug interaction contract studies in compliance with good laboratory practices, OECD principles of GLP, and Japanese GLP standards, and specializing in drug transport and drug metabolism studies. He became the technical support manager for services in 2020 and provides valuable guidance to ensure research needs are being met. Now, without further ado, we'll turn over our time to our first presenter, Dr. Scott Boley.
Thank you, George, for that kind introduction, and thank you to XenoTech for partnering with Sinclair to bring you this webinar. So I've worked with literally hundreds of companies over the years on various programs, and one of the common themes that come up, questions that come up are, how do I know which species that I'm going to use for my particular program? And on the side of the screen there, we've got the true heroes of biomedical research, the animal models that we use. These represent the primary species used for toxicology: rats and mice for the rodent side of things, dogs, monkey, and mini pigs for the non-rodent, and then rabbits are used to a fair degree as well. But these are the species that you will need to use in your non-clinical program. But how do you know which one's right for you? So we've got 6 that we commonly use. There's other species that can be used based on particular programs. They're more specialty. I've seen programs with sheep, etc. But these are the 6 that are most commonly used. As with anything, not everything's perfect. The best example here is, if you have to use mice, you're going to use a lot of mice because there's not a lot of blood to go in or to go around for the various parameters you're going to look at. So each have their own concern. But I guess one of the things that I've been hearing a lot more about first is a growing concern about the use of primates. There are people that are looking to not use primates for their products. It's more emotional than anything, I think. And in coupling with that, it's not just primates, it's also dogs. Recently, I've heard a growing concern about using dogs in non-clinical research. But as I said, it's more emotional or animal rights based. Your primates are close to humans and dogs are pets, so we don't want to use them for non-clinical research. But the driving factor that I would like you to come away from this webinar with is that we are in the business of toxicology, non-clinical research, because we are building a safety profile for a particular program, for a molecule. And that safety program is going to help protect the health of human volunteers and human patients in the clinical trials that you will have ongoing. So selection of your species is something that should be based on scientific rationale, not emotional feelings or anthropomorphizing, but scientific rationale, because you need to make sure that you're providing the best safety for the people that will be taking your drug. So how do you know? And tongue in cheek here, this is the universal answer of a toxicologist. I give this answer at least a dozen times a day, is that it depends. Because there are no one magic bullet answers to anything in toxicology. It depends on what your situation is. So first of all, we have guidance, lots and lots of guidance out there. The two that stand out when it comes to species, though, are the ICH S6 for biologics, your proteins, antibodies, etc., and then the ICH M3 for the small molecules. Now the S6, that covers it pretty good as far as how do you know which species is right? What's a relevant species? The M3 less so, but that's what we're here today to help you understand better. It's how do you know for a small molecule what species is right? Then, because everything is complicated, if you have a particular dose route, and the best example of that is if you're working in the dermal space, you're going to need to run a mini pig study. And the reason for that is because the skin of a mini pig is the best model for the human skin out of all the species that we use. So it's not just guidance, it's also what is your program, what particular area of therapy are you working in that can help guide which species is the right one for your particular program. So I'm going to touch upon biologics briefly, simply because they are a major class that's in the pipelines today. For species, it's all about the pharmacology. Can my test article, when given to an animal, elicit the desired pharmacological effect, yes or no? Now, to determine that, typically it's a tiered approach. So biologics are highly specific. They're designed that way. So the first thing you need to look at is what is the conservation of the sequence? Is it highly conserved down to mouse? Or is it 90% at primate, and then at the rodent level, maybe it's 30 or 20%. Using the conservation as kind of a first tier approach, then after that, if it's fairly conserved, you can look at in vitro or in vivo receptor binding and engagement. So say there is a decent amount of conservation in rat, well, does that mean that my test article will bind and actually elicit the desired effect? Yes or no? These are things that you do before you start doing any type of non-clinical animal studies. And what you'll find is that many times for biologics, primates are a model that you'll need, and a lot of that is because of the specificity of these particular test articles and a lack of conservation. But I've run many programs that also involve rats because the conservation for that particular target is fairly well conserved. So the rest of this talk is going to be focused on small molecules. 10 years ago or so, when biologics first started really ramping up, there was a lot of talk about how it was going to take over the market, and it would be the majority of the drugs in development would be biologics. But I can tell you from the CRO world, I still see a tremendous level of small molecule work going on. So it is still a major class of test articles for various companies. And a lot of times people will default to the rat and dog, and that's for good reason, because historically that's been shown to be what is needed for small molecules, But again, as I said earlier, it is not a guarantee that you cannot just use rat and dog and assume that it's going to be the right species for you, because things can be different. So how do you know? Again, just want to reiterate that there are some efforts to reduce or eliminate primates and dogs, but you need to approach this scientifically, because as a company, if you're developing a drug, you want to make sure that your safety profile provides the best assurances for the people that are going to be in the trials, that everything is, as much as you can, and it's as relevant as it can be, using a scientific approach in selecting which species you'll use for your non-clinical studies. And then for biologics, as I mentioned, it's about pharmacology. With small molecules, it's about metabolism. How is my test article broken down once it's administered into an animal? So some of the preliminary work, and you'll hear much more about this in the second half of the webinar, you can do some in vitro approaches using microsomes and hepatocytes. And these are things I've told many, many sponsors over the years. You look at human, mouse, rat, dog, mini pig, and primate. Look at the human profile, and then you choose the rodent and non-rodent that cover the human. Most of the time, it will be rat and dog. However, you will have occasions where the rat has some metabolite that is spiking really high that isn't present in human, or human has a metabolite that's not present in rat. So then maybe mouse is the right species. And same when you're looking at dog, mini pig, and primate. It doesn't always default to the rat and dog, because things are different. Your molecule is different. How it's metabolized can be unique to your particular program. But sometimes the profiles will be comparable. So you run the in vitro metabolic profiling, and rat and mouse are basically identical, and they both cover human. What then? So looking at things like pharmacology, so similar to the biologics, is the pathway that I'm impacting, is it more represented in the rat versus the mouse? Perhaps, it depends on your program. Is there any particular sensitivities that I need to be aware of for my particular test article? Then, of course, there's always, you can do some non-GLP PK studies. Take a look at your exposure profile. Because if everything's equivalent between rat and mouse, but the mouse has a better exposure profile, then that can be your justification. But again, this is all scientifically based on data that you're acquiring on your molecule. So I think I've said this a number of times, but it needs to be based on science. We are in the realm of, we're in the area of safety data, and we need to make sure that it is based on a rationale that has some data behind it and can provide the best safety for human beings. Now, I've said this to many sponsors over the years about the metabolic profiling, and I make it sound relatively easy. I say you do metabolic profiling, it'll help you decide what it is. But it's not a straightforward process. There's a lot of things that go into this. There's microsomes, there's hepatocytes. Which is better? Is one better than the other? Or is it basically a wash? Do you have to always include all the species into the microsomes or the hepatocytes? Or can you look at some of the species? So basically, just how exactly is it accomplished? I've recommended it to many sponsors, and to be honest, I've not known a lot about what goes into the metabolic profiling. And that's part of the reason that I feel that this webinar can be very useful to a lot of people. Because what we're going to do now is we're going to transition to having Andrew take over, and he's going to tell us more about how does this happen, so that when you're placing work with a CRO such as XenoTech for these kinds of studies, how is it that these assays are actually done, and what do I need to know so that I can be more informed as I move on? So at this point, I'm going to turn it over to Andrew.
Hey, Scott, thank you very much, and a great presentation and a really good overview. Yeah, what I'm about to talk about follows up really well with everything you said, and it's using in vitro drug metabolism and in vitro assays to help assist in the species selection for the non-clinical tox studies in the drug discovery process. Just a quick overview of what I plan to cover today. First, I'd like to just talk about the various test systems that are used for in vitro studies. as Scott was just mentioning, hepatocytes versus microsomes versus other subcellular fractions and what's used. And just to clarify too, between the in vivo tox work and the in vitro ADME DDI work that we conduct, we both use the word test systems, but have different meanings. For us, test systems refers to hepatocytes or subcellular fractions. And in the in vivo world, it of course refers to the species that's chosen for the test. So we'll talk about the various test system, what's available and the advantages of each and when you might choose one over the other. We'll then go into some of the in vitro study types that are available to help in selection of your non-clinical tox species. And these are stability, metabolite characterization, and enzyme induction. Okay, the various test systems for in vitro studies. The two main test systems or two main categories of test systems would be hepatocytes and subcellular fractions. So hepatocytes, of course, are intact living cells. They have all the metabolic pathways and everything's working. Subcellular fractions, on the other hand, are parts of cells. So the cells are ground up and spun down to separate them into their various fractions. Each fraction contains different enzymes and drug metabolizing enzymes that are present. So there are two main types of primary hepatocytes that can be used. These are suspended hepatocytes or attaching plated hepatocytes. And there are cases where you choose one over the other. For example, suspended hepatocytes are kind of the go-to. We try to use those the most, but they're limited in how long they can be incubated and remain viable for. Typically, suspended hepatocytes are used for shorter incubations, between 4 and 6 hours. Where on the other hand, attaching and plated hepatocytes are able to be used for much longer incubations. With media changes, attaching a plated hepatocytes can go 5 days or longer in incubation, again, changing the medium. However, there is the loss of some of the metabolic competency of the attaching plated hepatocytes over time. But when dealing with really low turnover compounds, it can be the proper test system to choose, and for other study types, such as enzyme induction. So suspended hepatocytes are used for metabolic stability studies, metabolite characterization studies, and being used for shorter incubations and coming at a slightly lesser cost. They're a good choice for medium throughput screening type studies if there's several lead compounds that are being evaluated to pick a lead. Again, the attaching and plated hepatocytes are used for enzyme induction studies, also metabolic stability studies, and metabolic characterization studies. Specifically, those last two study types are used for the low turnover compounds. And again, hepatocytes are kind of considered the gold standard of what to choose when conducting these in vitro metabolite studies, just for the fact that they are intact living cells and have all the metabolic pathways in operation, for phase 1 and phase 2 metabolism, and it just gives a more complete picture. Now, these hepatocytes, cryopreserved hepatocytes is the format that tends to be used most often these days, since they can be stored and used on demand. There are numerous non-human hepatocytes that are available. Of course, all the non-clinical animal species that Scott was just mentioning, dog, rat, mouse, monkey, rabbit, mini pig, hamster. So all these are available. Generally, when we use these, they're pooled, again, from the clinically relevant species, and having a really large pool, 50, 100, 200 donors in the pool, gives a much better representative picture of the metabolic profile of that species as a whole, as it averages out any potential individual differences that might be there. Working with these large pools also provides long-term lot availability and reproducibility. So if you do an experiment with one lot this year, chances are next year you can get the same lot and be able to have the comparison and the reproducibility. Again, these are used for the in vitro metabolism studies. This last point applies to both hepatocytes and subcellular fractions, but any in vitro test system that you're using for these drug metabolism studies should make sure that it's a characterized test system, so that the metabolic profile, at least for the major drug metabolizing enzymes, such as cytochrome P450s, have been characterized. And this allows you to then run a probe substrate in positive control and just make sure that the test system is competent and functioning as you expected it when interpreting your data. So subcellular fractions is the other main group of test system that are available for in vitro studies. And again, these are fractions of cells where the cell has been basically ground up and split into its different components. And they can come from many different tissues and many different species. Some of the main tissues that are requested for subcellular fractions, of course, are liver and intestine and kidney. Those are some of the big ones. But other tissues are available, such as skin and many other different tissue types. And again, these can come from many different species. Now, when you might choose a different tissue type over just liver is if your drug is intended to have its action at a particular site, or you think that the drug is going to be metabolized in a different tissue type, it's probably good to look at that tissue and look at the metabolism of your drug in that tissue along with the liver. There are different types of subcellular fractions. As Scott mentioned, microsomes. Microsomes are one of the go-to standards when it comes to looking at drug metabolisms. Microsomes are pieces of the endoplasmic reticulum, which includes the cytochrome P450 enzymes and also other enzymes, drug metabolizing enzymes such as UGT. S9 is another subcellular fraction. So S9 includes microsomes and cytosol mixed together. And then lastly, cytosol is another subcellular fraction that can be used. And again, you'd pick these just depending on the properties of your drug or the questions you're trying to answer, or if you think that any particular enzymes might be involved in the metabolism. If you have any foreknowledge of that, then you might choose one of these test systems, either in addition to hepatocytes or just one of these. So one thing about subcellular fractions, however, is since they are not living cells, many of the enzymes require cofactors to become active. For example, NADPH has got to be added to the subcellular fractions to activate the CYP enzymes, or UDPGA for UGT enzymes activity. Now, one neat thing about this, though, is that the enzymes can be turned on or off by the addition or omission of these required cofactors. And depending on the drug in question, its structure, and its putative metabolism, this could be an advantage of being able to assess what enzymes are involved or which ones aren't, and turning them on and off as required. Okay, now to get into some of the in vitro drug metabolism studies that are used to assess species comparison and help selection of non-clinical tox species. The first of these is pretty simple, and it is a metabolic stability study. So again, typical species that are used in these study types would be human, rat, mouse, dog, rabbit, monkey, pig. And here's an example, some example data from a study that was done comparing rat, dog, monkey, and human. And all these plots are the same, where the y-axis is the percent of the drug that's remaining, just plotted against as a function of time, which is on the x-axis. And in this example here, you can see that rat and monkey are most comparable to human when it comes to their half-life and intrinsic clearance, where dog has a much faster loss rate or clearance of the drug in question here. So in this case, using this data, you might start to consider rat and monkey for the non-clinical tox species. The next in vitro study type in drug metabolism that's used for interspecies comparison would be Comparative Metabolic ID, or to use more precise language, metabolite characterization. Now, the goals of the metabolite characterization study are to compile a complete profile of all the metabolites that are formed when the drug is metabolized. And this is done to find out what other species have a similar metabolic profile to human, and also to look at, are there any human-specific metabolites that are formed, or are there any human disproportionate metabolites that are formed? You know, as Scott mentioned, and as we all know, different species might have different metabolic pathways or metabolize the drug in a different way. And one example of that is coumarin, where in humans, coumarin is metabolized to 7-hydroxycoumarin, which is not toxic. However, in rats, the coumarin is metabolized to an epoxide, which is very hepatotoxic. So knowing this information up front, again, allows you to select the appropriate species and will assist with that. Now, one thing to keep in mind and that I want to mention is that metabolite characterization is done using mass spectrometry. And for the most part, metabolite characterization is a qualitative assay, not a quantitative assay. The reason for this has to do with the differences in ionization efficiencies for different metabolites or different parts of the drug as it's metabolized. And it relates to the ionization efficiency and not exactly to how much of the drug is actually present. Now, these study types can be done quantitatively, and there's a couple different ways for that. The first is if a metabolite reference standard is available. So you can run a calibration curve and then, of course, quantify the amount of metabolite that's formed. However, when these studies are generally first done, you know, we don't know what metabolites are going to be present, so it's probably highly unlikely that a reference standard is available. Another way to make the study quantitative would be to use a radio-labeled parent compound. And if we have a radio-labeled parent compound, and assuming that the radio label stays on the metabolites, then we're able to quantify the metabolites. Lastly, if the metabolites have a UV signal and are detected in UV, that's more semi-quantitative. So it's a good middle ground, and all these study types are generally run with a UV detector. And using the UV data, you can make the semi-quantitative comparison between the abundance of metabolites that are present. Now, that being said, even though it's not quantitative, generally, you can make a comparison between species for a given metabolite. So metabolite 1 was higher in humans than it was in rat. However, you just can't make a comparison between metabolite 1 and metabolite 2 or further metabolites within a given species. And just an example of some of the data that would come out of a cross-species metabolite characterization study would be a table like this, where you could see all the rows of the different components or metabolites that were formed from these various species. See the retention time and the mass shift of the molecule. And using this mass shift, we're able to provide putative biotransformations that happen to the molecule. And then in this example, you can see that metabolites 1 through 5 were formed in all species. However, metabolite 6 was only formed in pig and human. So again, in this case, pig would be the more appropriate non-clinical test system to use in the tox studies moving forward. Okay, lastly, I'd like to mention enzyme induction studies that are sometimes done to do cross-species comparison. And there's 2 ways that these can be done. The first is in vitro, and there's also an ex vivo study design that can be conducted. As you all probably remember from our basic biology, enzyme induction is a process in which the drug will bind to a nuclear receptor and trigger transcription of a given gene. You know, in this case, you're looking for transcription of drug metabolizing enzymes. Now, since it takes time for the enzyme to be transcribed, translated, and turned into protein, Enzyme induction studies, in vitro enzyme induction studies, require a lot more time. And they also require, or the only test system that can be used for these study types is hepatocytes, since you need an intact functioning cell with all the cellular machinery operational to be able to do that. So these are done in plated hepatocytes, generally with the test article treated for 3 days in order to see if there's any induction potential of the drug in question. One benefit to in vitro induction studies is it only requires a very small amount of test article, so milligram amounts. And again, this can be done in mouse, rat, dog, and monkey. The endpoints for enzyme induction study is either messenger RNA or enzyme activity. The preferred one out of these two is actually messenger RNA, as it's cheaper, faster to do, and it's also preferred by the regulatory agencies when conducting these as part of an IND enabling study. And there's some reasons for that. If you'd like more details, I'd point you to our website, and we have more information as to why that is. And lastly, for enzyme induction studies can be conducted ex vivo, where the induction is looked at in the laboratory animals, typically done following a tox study, again with the main species. So the animals are dosed by the sponsor or the CRO conducting the tox study, and the liver and tissues samples are sent out for analysis. And again, the same endpoints are looked at in an ex vivo study, be that messenger RNA or enzyme activity, to assess the induction of the enzymes in question. One drawback to doing it this way is that larger amounts of test article are required, and of course, depending on the tox species, you're going to need more for a dog or monkey than for a mouse. If it is part of a tox study or a follow-up to a tox study, it's generally not an issue because the test article has already been manufactured and it's there for use in the study. But sometimes this can be an issue if an enzyme induction study ex vivo is going to be done just on its own, maybe before enough compounds available. So just something to keep in mind. So one question, again, with all of these study types, be they in vitro, ADME, DDI work, or non-clinical toxicology studies, is why conduct these studies? Is this just for box checking? And our answer to that is a really strong no. The information in aggregate that comes from all these studies actually has a lot of real utility to the sponsor. For one, it provides a deeper understanding of the molecule. It gives you insight into the metabolism of the molecule, the enzymes involved in its metabolism, and the information that's generated from these DDI studies, be they in vitro or in vivo, is the information that's going to go on the drug label and keep the drug safe. From the pharma company's perspective, these studies, in vitro and in vivo upfront, when conducted early, can inform go and no-go decisions for a drug candidate. So they can bring a lot of information on that when looking at drug candidate selection or, making those decisions. And the predictive toxicology and dose selection for future non-clinical in vivo studies can help be determined by some of the in vitro work that's conducted beforehand. Second, it prepares for clinical studies. The work done in the tox studies and the in vitro studies can help give a prediction of what first-in-human dose will be, and if there's going to be any potential drug-drug interaction risk that'll help for the design of the clinical study. And lastly, and of course, it satisfies the regulatory expectations and allows the sponsor to comply with the regulatory guidance. So while individually and on their own, each one of these studies may appear deceptively simple, maximizing the insight that can be gained from all of it and putting that data together in an aggregate and getting all that utility can be a complex process, but a very important one to undertake in the drug discovery process. So just to summarize everything that we talked about today, as Scott mentioned, and I agree with Scott wholeheartedly, It's very important that species selection be based on scientific reasoning, and that should come from looking at good scientific data that gets you there. The decision of what species to use should also be pharmacologically based. Choosing the right test system or right species to match human as closely as possible. That being said, choosing the right test system is very important. Be that which tox species to use for in vivo, or which type of in vitro test system, hepatocytes, subcellular fractions, what type of subcellular fraction for in vitro work. Putting that thought up front, knowing what questions you need to answer is invaluable, and then choosing the right test system to do that in. In vitro species comparisons are very useful to help before you go into the in vivo work. Looking at compound stability, metabolite characterization, and enzyme induction as a screening study up front, or as a follow-up to in vivo work as needed. And lastly, just to always remember that these studies are more than just box checking, and this information has real utility. Never lose sight that these drugs are going into humans, so we need to make them as safe as possible and do as much testing as we can. And on that note, I think we're going to open it up to questions.
Great, thank you, Andrew, and thank you, Scott, for the engaging presentation. Does enzyme activity supersede the mRNA fold induction? Let's say you get about 50-fold increase in mRNA expression and far more than 20% of the positive control, but no increase in activity and no inhibition then in the enzyme activity. Should convince the regulatory that there is no induction issues. Is that correct?
Yeah, no, that's a great question. So how should I start this? As I mentioned, the preferred endpoint is just mRNA. Now, sometimes clients will do both, and that's always great because it gives your data set, makes it more robust and gives you more insight. But yes, what do you do when you have conflicting results like that, where you have mRNA induction, but you don't see any change in the activity? One of the reasons for that, and one of the reasons that mRNA is the preferred endpoint, is that there's the potential that the drug or one of its metabolites might be inhibiting the enzyme that's being induced, and therefore you could have induction, but looking at activity, the induction might be masked. Now, in that question, I think it asked if there was no inhibition of the enzyme, but you still saw the fold increase. The regulatory agency and the guidance is pretty clear that even if there's just one of them, it's going to be considered an inducer, and further work's going to need to be done. Either look at it through the static or mechanistic models, or follow up with clinical work.
Great. Our next question comes in from Bill. Since only major human metabolites need to be qualified in your animal species, do we really need to be concerned if your animal species does not cover a particular low-level human metabolite?
Good question. I can answer it from the in vitro side. Again, the guidance has some pretty clear statements that when metabolites need to be considered. If it's a major metabolite, say 25% greater area of the curve than the parent, or if it's a toxic or reactive metabolite, then they need to be assessed. So if it's a really minor metabolite, probably not. I don't know if Scott has anything to add to that from his side.
Yeah, like you said, if it's based on AUCs, you do have to track some of them per the guidance. But the big unknown there is exactly what you also said. Is it a reactive? What's the structure of it? So it might be a minor peak, but if it's a bad actor, then it kind of changes the landscape. But for the most part, I would imagine that if it's kind of in the noise, it's not really of concern.
Okay, Moran asked, is it important which strain of rodents is used? And is it possible to switch strains between repeat dose studies in order to test the toxicity in different strains?
Andrew, you want to handle the in vitro side and I can handle the in vivo side?
Yeah, I mean, from the in vitro side, I personally never ran across that. You know, generally we just use a particular lot, and then if the client comes back, that they'll request the same lot that's already been in use, which is one great thing with having such big pools and big lots to choose from. So I don't have much to say from the in vitro side.
So if you started with, say, Sprague-Dawley and you wanted to change to Wistar, it does carry risks. I know of an instance where we actually used Sprague-Dawleys from vendor A and Sprague-Dawleys from vendor B and got a different result. So even though they are the same strain, well, not suppose, I mean, they are the same strain, there is genetic drift because these breeders have kept them separate from the other breeders for generations. So there has been some drift. So I know people that switch between strains, but it's something that should be done cautiously. Typically, when you start with Wistar or Sprague's, you tend to stay with them for the duration of your program. So you can do it, it's just risky.
And Matthew asks, can humanized mice play a role in preclinical tox testing?
So now we're talking more along like the disease models, and that is up and coming. But again, there are concerns with that, and the answer is it depends. Because with those models, what we are lacking is a historical database with like microscopic findings. So if you see, some type of an odd finding in the adrenal gland, does it mean it's your test article, or is it something that's common to this particular model? This will be, addressed as time progresses because more people are using these models of disease and alternative models for various reasons, and they're good scientific reasons. But the lack of the historical data can make it challenging based on what you're finding. If it's clean, then you're pretty good. But if you have one of these weird kind of findings, then it can raise some questions that you'll have to deal with the regulatory bodies.
Our next question, for in vitro screening, do you need to use both rhesus and Sinnoh strains when you're screening for the NHP? And similarly for mini pigs, does it matter which strain?
Yeah, good question, and I'll answer my part, but then I'm going to kick it over to Scott because on the in vitro side, there's nothing really in the guidance that specifies what strain that needs to be used. So from our perspective, I'd say no. But I don't.
And yeah,
Sorry,
from
Scott.
the tox side of things, I mean, most primate work is done in Sinnohs. That's where the historical data is. And as far as pigs go, same thing. I know there's a lot of people that feel like Gottingens are the gold standards, and they have been used for a long time. There's a lot of data on them. But I also know that if it's a Sinclair or a Hanford or a Yucatan, it's still accepted by the regulatory agency. So depending on what your program is, you're not tied into any one strain. I mean, I guess the caveat there is on the primate side, it could be. I mean, there could be things that are specific to a rhesus that don't really cross-react in a Sinnoh, and I've run across those programs where it had to be a rhesus. But for the pigs, I don't know of anything that says you have to use A or B versus the other strains.
And then Boris has another question on humanized mice that you may have already answered in your previous response, but is it beneficial to run studies in humanized mice to match the human metabolism profile?
I don't know that much about metabolism data in these models. I do know that the animal models of disease, these transgenic animals, do have some attractiveness because, I mean, typically we're dosing healthy animals. But in the clinic, you're not dosing healthy humans. So there is some merit and some good science behind using an animal model for disease. But as I said, you run into the whole, background data thing. As far as metabolism, I don't know that much about these humanized animals to know whether or not they truly represent like a more human-like metabolic profile.
Our next question, how reliable is the metabolic profiling when compared to in vivo studies?
Yes, that's a great question. and a little difficult to answer. So I don't have any statistics on like how often there's like a missed comparison between an in vivo and in vitro study. That being said, you know, it's well known in our industry that using these test systems, hepatocytes or microsomes, gives a really good foresight onto what metabolites are going to be formed. You know, these test systems and this type of in vitro work has been done for a really long time, studied well. There's a lot in the literature, and it's accepted by the regulatory agencies. So with that said, yeah, I'd say it's pretty predictive, and it gives you really good insight onto what's going to happen. Is that always the case? You know, absolutely not. There is a difference between in vivo and in vitro. And sometimes you'll see something different in the clinic, in your non-clinical tox species that you didn't see in the in vitro work. So, you know, it's complex, but I think it's a very good start and gives you a lot of insight. Do you have any follow-up to that, Scott?
No, I guess a question for me would be, you know, I always tell people that rat and dog cover most of the small molecule, and you're the one that runs those assays. And would you agree that rat and dog for small molecule is like 80 to 90% of the time, but not always?
Similar to human or similar to
No,
in vivo?
That rat and dog are the appropriate species based on in vitro metabolic profiling.
Yeah, you know, it does tend to be that way. Again, I don't have any hardcore statistics. Now you're making me want to go look that up with the historical data. But yeah, no, I would agree with that assessment.
Okay.
So how often is the non-rodent species something other than dog? Is it something like 20%, the difference there, Scott?
In programs that I've seen, and I'm assuming sponsors have already done their metabolic profiling before they get to me, it's 20% or less that it's going to be a pig or a monkey.
Then what's the appropriate time in your research program to perform metabolite identification or screening tests?
I think as early as possible. You know, there's so much good information that comes out of that. And, you know, if one of your metabolites is some toxic or reactive metabolite that's going to, well, potentially kill the project, that's probably something that you want to know as early as possible.
To follow up to that, how long do the studies take? And do you need to do all of them?
That you cut out on the last part. How long do they take? And
do you need to do all of them?
And do you need to do all of them? Great question. You know, on average, you know, running these studies start to finish, you know, not counting lead times, it's probably 6 weeks, 8 weeks. For the metabolite characterization studies, although a lot of that can depend on how many metabolites are formed. You know, if it's only a few metabolites, it's pretty quick. If there's 20, 30 metabolites that are formed, it's going to take a lot longer, you know, to go through that data and look at the, you know, what the biotransformations are and things like that. Do you need to conduct all of them? It's probably a good idea, at least to do the MET-ID, metabolic stability. You know, a lot of the other ones, because there's a bunch of different study types, they are required by the guidance. So, you know, following up with human enzyme inhibition studies and human enzyme induction studies, drug transporter studies. So you know, I think at the end of the day, with the compound that's going forward, eventually they're all going to get done. It's just a matter of at what stage and timing for it. And stage and timing for it, like Scott says, there is no answer. You know, it depends. It depends on where the client happens to be within their program, you know, how much de-risking they want to do up front as opposed to on the back end. So, you know, there's a lot of things that can determine how that works.
Okay, and I've got a final question here. If anybody else has questions to pose, please submit them now. Pradeep asks, is the species selection always scientific decision, or practical feasibility and ethical aspects also play an important role in this? And does it apply for any specific study types or molecule types?
What I've always advised sponsors at the, because I'll have sponsors say, well, I don't want to use primates because they cost too much. And the FDA doesn't care how much it costs. The FDA cares about how relevant the model is. So if you try to submit a program and you say, I don't use dogs because I like dogs, that's probably not going to be a good idea and probably won't go very far, which is why I said, it's about science. And I understand the ethical concerns, the emotional concerns. I own 2 dogs. I love dogs, but I also know that, I need to work to provide the best data so that the people that will be taking the drug are protected as best they can be. So it's a fine dance, but I think that if you go with science, you can stand on science. And I don't think the FDA can really argue with that.
Well, thanks, Scott. With that, it looks like we've covered all of our questions. Scott, Andrew, is there anything else you wanted to cover before we wrap up this webinar?
I'm good.
Yep, no, I'm good. I appreciated doing this. This was a great experience and a great webinar I think we put on. So thanks, Scott and Sinclair team.
Yep. Thank you, Andrew.
Thank you again for joining us, and we hope to see you again at our future webinars.